Welcome to another Study With Me video, Microbiology edition. First off, if you haven't subscribed, hit the subscribe button. Also hit that bell so you'll get notified when my next video is posted. If you have any microbiology questions, feel free to post them below. Now, if you're interested in joining me in my microbiology study program, my email's in the description box and I'll be happy to give you more information about that. I also teamed up with Check Tutors, so if you need personal help, feel free to find me on there and I will help you. So let's get started in this video. It's about inhibitors, non-competitive and competitive inhibitors. Inhibitors. Hinder, restrain, prevent, block, stop. These are what inhibitors do. Today we're going to learn about competitive inhibitors and non-competitive inhibitors. So here we have our beaker, and in that beaker we're going to place substrates. Next we're going to add in our enzymes. Now what the enzymes do in the presence of substrates is that they bind and form an enzyme substrate complex. Now after they're binding, the substrate alters its shape but the enzyme, its shape does not change. It binds to another substrate. So what happens if we add a competitive inhibitor? Well, they too can bind to the substrate. So if the competitive inhibitor binds to the substrate, well, what happens to the enzymes? They cannot bind to the substrate. So the purpose of the competitive inhibitor is to slow down the enzyme substrate complex. How can you combat reversible competitive inhibitors? Well, add more substrate. If there's more substrate in your beaker, then the enzymes have a better opportunity to make enzyme substrate complexes. So here we have an example. We have our substrate PAVA, which is para-amino-benzoic acid. We have our enzyme and our sulfur drug, which is our competitive inhibitor. Our sulfur drug here is sulfanilamide. So our substrate binds to the active site of the enzyme, thus producing folic acid. Folic acid is a coenzyme that is needed for bacterial growth. So what happens when the competitive inhibitor is introduced to the mix? So no substrate. We add our inhibitor, the sulfur drug. The inhibitor binds to the enzyme, but then makes no folic acid. So there's no binding of the substrate, no folic acid. Non-competitive inhibitors do not compete with substrates for the active site, but has another site on the enzyme called allosteric site. That's here. So the non-competitive inhibitor can bind to the enzyme. Well, what happens to the enzyme? Well, the enzyme changes shape, thus changing the active site. Now the substrate cannot bind to the active site, leaving the substrate to be alone. Now this whole process is called allosteric inhibition. It's when the non-competitive inhibitor binds to the enzyme at the allosteric site. This changes the shape of the enzyme and not allowing the substrate to bind to the active site. Now, can this process be reversed? Yes, in some cases it can. So the non-competitive inhibitor can leave, allowing the enzyme to go back to its normal state or normal shape, and the substrate can then bind to the enzyme again, making the Now you have all of this information down, whether on paper or in your brain. So now that it's in your brain, you have to bring it forward. So we're going to begin with a substrate. You have a beaker full of substrate and you add an enzyme. What happens? What's created? What's produced? Now you have your substrate, enzyme, and a competitive inhibitor. What happens in this scenario? Next scenario, you have your substrate, you have your enzyme, and you have your non-competitive inhibitor. What happens in this instance? Okay. Now we have an enzyme. What are the two sites that either a substrate or a non-competitive or competitive inhibitor can attach to? Well, thanks for watching my video. Don't forget to hit the subscribe button and that bell so you get notified for the next video. If you have any video ideas, comment below. Any questions, comment below. Don't forget, I do have a microbiology study program, so if you really need that personal, in-depth help, feel free to email me. Again, my email's in the description box. Also, I'm on Check Tutors, so if you want some one-on-one time with me and need that help in microbiology, feel free to find me there. And I'll see you in the next video.